we've heard a lot at this meeting about various efforts to target ALL-associated antigens with chimeric antigen receptor T cells. What I wanted to talk to you guys today about in a short story is our work really in the AML CAR T cell field, and specifically with trying to kill these T cells in an attempt to mitigate toxicity. Okay. So I don't need to remind this audience that we have a long way to go therapeutically in terms of curing children with AML. When we consent families, we usually say that we cure about 60% of patients with newly diagnosed AML the first time around, and this is with very intensive multi-agent chemotherapy as well as often with hematopoietic stem cell transplantation. And I think, like ALL, we've learned a lot about risk stratification of these patients by underlying leukemia genetics, as well as their responses to therapy in terms of minimal residual disease at the end of the first month of chemotherapy. But for some patients, we really still have a long way to go. A 25% event-free survival at 3 years is pretty terrible. And we know that relapsed AML today remains a significant source of childhood cancer mortality, and we are unfortunately often unable to salvage some of these patients, even with very intensive reinduction regimens. And unfortunately, in the last 4 to 5 decades, there have been very few new drugs for childhood AML that have been successfully brought to fruition. And in the last 10 years or so, there have been tremendous ongoing efforts, I think, with the evolution of next-gen sequencing techniques and a lot of technologies that have become a lot more affordable to really identify AML antigens for therapeutic targeting with a variety of small molecule inhibitors or other strategies, such as signal transduction inhibitors, other small molecules, epigenetic targeting agents, and antibody or adoptive cellular immunotherapies, and I'll talk about our work with the last here. So we demonstrated last year, and I'll thank Dr. Wigginton for introducing CD123 as a target in AML. Again, this is the IL-3 receptor alpha chain, and a lot of groups have focused pretty intensively on targeting CD123. But we demonstrated with a lentivirally transduced second-generation CAR, this is the PEN backbone, targeting CD123, that animals could be long-term survivors, could cure themselves of their leukemia, which is really what we think about as an on-target, on-tumor effect. However, we were pretty surprised to see, and I think unlike perhaps what we heard about today with the DART experience, our CD123 CAR T cells actually resulted in profound myeloablation when we tested those T cells in normal human hematopoiesis mouse models, or what we call humanized immune system mice. And this has been, I think, very perplexing to us, and we haven't entirely figured out what that mechanism is. We know that our T cells are persistent, and so they probably go on to zap any 123 positive cell, and we hypothesize that perhaps with forced maturation into myelopoiesis with these T cells sticking around, that they're just really whacking any 123 positivity that might evolve. But I think, as Crystal and many other people highlighted in this meeting, there is no perfect antigen for cancer, including AML, and we think that any antigen that we may target in AML is going to result in some sort of hematologic toxicity. And so what we're really worried about, too, is that with targeting AML with CAR T cells, we may actually need to go on to transplant these patients, given this pretty significant risk of heme tox. And so the fundamental question of this line of work with this project is, can we terminate these T cells after some sort of, in this case, CD123 redirected CAR T cell induction of leukemia remission? Is there a sweet spot that we can target and we can turn these T cells off such that we can preserve leukemia remission? And I think our clinical experience, and you've heard a lot about this with targeting CD19 in patients with ALL and other B-cell malignancies, is that some of these CAR T cells really stick around for a long time, whether it's 63 days, whether it's years. I think it really depends on the experience that we've had. But we think that this is going to be particularly problematic for patients with AML. And so our hypothesis is that T cell elimination at an optimized time point could increase the therapeutic window for using a CAR T cell-based strategy for AML. And so there's various T cell termination strategies that are under study by our lab, and I think you heard a lot about that from Mike at this meeting as well. But we have been looking at biodegradable CAR T cells. So these are T cells that are electroporated with tumor antigen, scFvs, so they stick around for only a short period of time because they're messenger RNA-based. There's various suicide switch ideas that you've heard about, like inducible caspase-9 or the truncated EGFR receptor that you can intercalate into the construct itself. And then some of the work that I'll talk to you about today is our efforts to look at T-cell depletion using monoclonal antibodies. And you can do this against endogenous antigens on the surface of the T-cell, or as well as antigens that you engineer onto the surface of the T-cell. And so what I'm going to talk to you about today is our efforts to terminate T cells using Alemtuzumab, and I don't think anyone in this audience finds Alemtuzumab to be a foreign concept. This is a humanized anti-CD52 monoclonal antibody. CD52 protein is highly expressed on the surface of lymphocytes, particularly B cells and T cells. There is lower expression also on monocytes, macrophages, granulocytes, and some NK cells. And it's not entirely known how CAMPath works, but we think that it is some mechanisms of ADCC and CDC. There's current clinical indications. I think a lot of us have experience with using CAMPath in our patients. It's currently FDA approved for adults who have CLL. It's also used, interestingly, in multiple sclerosis as an immunomodulating agent. I think also we have experience using this in some patients as part of their conditioning for T-cell depletion and some stem cell transplantation, as well as to salvage patients who have refractory HLH. The half-life in humans is similar to other monoclonal antibodies. It lasts about 4 to 5 days. The effects seem to be linked somewhat to the dose and also the duration of CAMPath administration. Interestingly, in patients who are treated with CAMPath, they tend to recover their B cells fairly quickly, within 2 to 3 months. But the T-cell lymphopenia can persist for months to even years. So we first, when we were starting this project, were curious to see, number one, does Alemtuzumab or CAMPath have any effect on AML cells themselves? We use a lot of monocytic origin AML cell lines, I think, commonly in the lab. And so we first just looked at this by flow cytometry, and we see on some of the cell lines a blush of CD52 expression. And so we first wanted to test this using our xenograft models in NSG or NSGS mice. Here, as you would expect, because this is a human monoclonal antibody, we see no effect in murine tissues in animals that are just treated with CAMPath. However, in animals who are engrafted with MOLM14 cells or other primary pediatric AML cell lines that we've used, we really see no effect after CAMPath in them either when we look at tissues after treatment with CAMPath about a week later. So this was very encouraging to us that whatever model system that we were developing was going to be pretty clean. So we went on to use our CART123 tool and tried to figure out, what was the optimal dose of giving Alemtuzumab as well as what was the optimal timing. And these are very small models, sorry, but these are bioluminescent animal models of human AML cell lines. We've also done this a lot in primary or patient-derived xenograft models. You can see here in animals that were treated, that were engrafted, in this case with MOLM14, treated with saline or our controlled untransduced T cells, the animals develop rapid AML progression and die somewhere around 3 to 4 weeks. You can see here with CART123, we see rapid clearance of the AML. These animals go on to clear their burden and become long-term survivors. We followed these animals out as long as a year, and they remain in remission. So this is probably a pretty potent T cell. When you look here, we can follow the animals by retroorbital venous bleeding. We can quantify the T cells in the periphery. And so you can see here, this is a very measurable output for us. But then we went ahead and tried to figure out, coming in with Alemtuzumab at 1 week, 2 week, 3 week, 4 week after giving CART123 to really figure out what that sweet spot was, what was the best timing. And you can see here, or maybe you can't see here because it's quite small, but a lot of these animals, when we came in with CAMPath, had not yet cleared their AML burden. And so when we went in and came to hit them with some Alemtuzumab, you see that the animals ultimately go on to develop rapid disease progression and they die with very similar kinetics to an animal that's just engrafted with AML themselves. But we did find, and so that was indirect evidence to us that we were probably depleting the T cells very carefully, with this AML reemergence. But the real proof of principle, I think, was in a relapse model. And so in animals who had been treated with CART123, who had no bioluminescent or no flow cytometric evidence of residual leukemia, when we came in with CAMPath, you can see here in animals that were treated with CART123 but not ablated with Alemtuzumab, we see a resurgence of the CAR T cells that's measurable in the peripheral blood. Those animals go on to clear their leukemia, so those T cells are sticking around, they're still functional, and they can stop up relapse. However, in animals that were CAMPath ablated after CART123 treatment, we see rapid disease progression, and those animals go on to die within about 2 to 3 weeks. And so that was further evidence to us that a single dose of CAMPath could actually kill T cells pretty cleanly. We see this even better, I think, in some of our primary pediatric AML Xenograft models. I'll editorialize for a moment that we've spent about 2 or 3 years developing pediatric AML Xenograft models. I think there's a very good reason they don't really exist. But it's been a very nice tool for us to evaluate novel agents, including CAR T cells. So this is a representative model. This is actually a transformed JMML into AML with monosomy 7, a very aggressive leukemia in our models. It's transplantable, so that's been a very nice tool for us. And you can see here in the gray and then in the black lines, those are our control animals. So these are animals that are xenografted with this AML specimen, treated either with saline or untransduced T cells as controls, or we come in with our CART123. And you can see here, these animals actually clear their disease very rapidly within about 2 weeks. We came in at what we thought was an optimized time point and gave alemtuzumab at 4 weeks post CART123. These are the blue and the red lines. So you can't really see here when we bleed these mice any difference. We've already cleared their leukemia from the periphery, so we weren't entirely sure what was going on, but we came ahead and gave this Campath at this 4-week time point. But you can see when we measure the T cells in the periphery, as soon as we give the Campath, we bleed the mice a day later, and the T cells are gone. So this is a very complete and very rapid response. And I think what was most telling is that when we looked in the end organs of these animals, and this is at a 4-week time point after giving alemtuzumab, so 8 weeks after CAR T cells, you see our control animals have a lot of AML replacement in the spleens, almost virtual placement in the marrows, but there seems to be no difference in the CART123 or the CART123 with alemtuzumab. So we seem to have found, at least in this model, a sweet spot of turning off those T cells when the animals are in remission, but the addition of Campath doesn't result in any disease progression. And so at least in these xenograft models, we think that this is a reasonable strategy. Okay. So I hope that I've shown you from these studies that in these models that one dose of alemtuzumab at a pretty optimized dose rapidly eliminates our CART123 cells in these preclinical AML models. We've done some additional in vitro experimentation, and it does look indeed that the mechanism of killing is ADCC with a little bit of CDC. We think in our xenografts that the sustained AML remission is best achieved by giving CAR T cells and then coming in to kill them at 1 month later, and that this elimination appears to be relatively complete based on some of the relapsed and re-challenge models that I showed you. I don't have time to show you today, but we've done a lot of work with the biodegradable CAR system using messenger RNA CARs. I will also say here, just in conclusion, that when we give these with sort of a prime and boost mentality, we give several doses of RNA CAR T cells. They only last in vivo about 48 to 72 hours. We can rapidly eliminate AML both in cell line and primary xenograft models, so we think that this is pretty cool too. But we do have some ongoing studies in the lab looking at the efficacy of alemtuzumab against some of the other AML CAR T cells on which we are working, as well as some of the other T cell termination approaches that I mentioned in the beginning. So I'll finish with just one last bit of clinical translation, and just to let you know that this has been a very, I think, exciting time to be in this field, and based on true bench-to-bedside translation, I think this is just a cool opportunity to highlight really the power of collaborative research in the laboratory. And we are very worried that given the myeloablative potential of targeting CD123, that this could be a very dangerous approach as we try to translate this to patients. We are planning to open a pair of phase 1 trials at CHOP and at Penn really in the next 6 to 9 months. And because of our concerns for myeloablation and because of our preclinical data with our RNA biodegradable cells looking really clean, we are going to start out first by using RNA CAR T cells. And so there will be a trial opening at Penn, we're hoping in the next couple of months actually, that is Novartis sponsored, but is going to be looking at the RNA CAR T cell against 123 for adults with relapsed and refractory AML. We are planning to have a pediatric phase 1 open at CHOP probably sometime in the spring or summer of 2016, once we get a little bit of experience, I think, to see how these look in the adults. Our dosing strategy is going to be 6 doses, again, because of the short persistence in vivo of RNA CAR T cells, kind of on a Monday, Wednesday, Friday, potentially outpatient schedule. We will give intercalated Cytoxan to kill the T cells that sort of remain but have extruded their RNA. This is a phase 1 trial, so there's appropriate dose escalation and de-escalation plans that will depend on patients' dose-limiting toxicities that we see. And I will point out for our initial trials that we are requiring that patients have a stem cell donor in hand at the time of enrollment on this trial, given our concerns that we may make these patients aplastic. So we are in the first trials planning to transplant our patients. So if we find that the toxicity is manageable, and if we see a little bit of efficacy or not, even in the context of a phase 1 trial, with our RNA CARs, we may consider testing the lentiviral 123 and potentially with an Alemtuzumab mediated depletion or using one of our other suicide strategies. So thank you for your attention. I'd like to acknowledge all the hands and the brains that are involved in this work. This is really a collaborative effort, both at the local CHOP Penn level as well as nationally with our Stand Up to Cancer colleagues, as well as the support of this Children's Oncology group. And I'll also acknowledge our very generous research support that has helped us support some of these hands and the brains, and also, I will say, has provided outstanding accommodation and health care for our mice. Okay, thanks.